All right, so I'm gonna have my dad on my TikTok today. His name is Bob.
Hello.
So, where did you go to college?
Oh, I went to the Jesuit College for my undergraduate, BS in chemistry. It was a little college called Fairfield in Connecticut. Then I went to Purdue for my PhD in chemistry.
What year did you graduate?
Graduated in 1986.
86. How long was your entire schooling?
Well, 4 years for BS and then 5 years for PhD.
And what was your thesis about?
It was about mass spectrometry and computer pattern recognition, kind of an early version of AI.
All right. What was your first job?
So from Purdue, I worked for an instrument company for a year, Brooker Instruments, out in Massachusetts. And then I got an opportunity to go to work for Eli Lilly back in Indiana.
How long were you at Lilly?
I was there for 27 years. So yeah, I left during downsizing.
What was the first drug you worked on?
I was hired at Clinton Laboratories in Clinton, Indiana to support Cefaclor, or C-Clor is the trade name. It's a cephalosporin antibiotic that was used for like ear infections, and it's very popular. It was a big seller back in the 70s, 80s. Once C-Clor went off patent, Lilly wanted to make a new drug. It was like it. It was still patent protected, but that wasn't, it was kind of a flop.
What was the flop drug?
It was called Lorabid.
Oh.
So yeah, YOUR.FAV.PHARMACIST.
YOUR.FAV.
It was basically a C-Clor molecule with instead of a sulfur in the ring, it had a carbon in the ring.
Oh. So it wasn't even a cephalosporin anymore.
It was a carbacefm.
Oh. What was the next drug you worked on?
Well, there at Clinton Laboratories, they also did animal health products. So I supported Monensin, Tylosin, and Nericin animal health products, growth promoters.
What are those?
Those were weekly ionophores. Feed them to the cows and the pigs and
What is an ionophore?
chickens. Ionophore?
Yeah.
It's like a macrocyclic polyether antibiotic. It would keep the animals healthy so they put on more weight.
I don't think we have anything for humans that's like that. I mean Zyprexa, but that's a different drug class. But ionophore.
Yeah, it's more like erythromycin, similar to that.
What's the drug approval process like for animal drugs versus human drugs?
It's probably pretty similar. Actually, the FDA is more concerned about animal drugs because, you know, a human drug like an antibiotic, you take it for a couple weeks. But an animal drug, they feed it to the animals, and you could eat the meat every day. So they're more concerned about residues, and so they're much more strict in terms of approval for animal drugs.
Okay, so animal drugs as in like
growth promoters.
So animals as in like food.
Exactly, food animals.
What about like animals like pets?
Yeah, I don't know that that's regulated by FDA. That's a different thing. Didn't really work much with that.
Because we didn't learn about that in school. We just learned about like INDA, preclinical animal trials, phase 1, 2, 3, NDA, and post-clinical surveillance. We didn't learn about that with like the animal process, so I'm not familiar with it. Where's the next drug you worked on?
So after I left Clinton Laboratories, we moved up to Lafayette, to Tippecanoe Laboratories. And then I worked on multiple compounds that were in phase 2 and phase 3 trials, drugs in clinical trials. Worked on one for BPH and some other things.
What was that drug?
Yeah, I don't remember. It was one that died in the clinic. I think maybe it had tox.
Was it like tamsulosin or was it like finasteride? Do you remember if it was an alpha-1 blocker or if it was a...
No idea, it's been too long.
It's been too long. Why did we move up like to like Carmel?
We first moved up to Lafayette because C-Clor went off patent. And then after the patent died, Lilly couldn't charge as much for it as they wanted because all the generics started to make it. So our sales plummeted, and we basically ended up closing that human health facility down. For Lorabid, they invested like $500 million in manufacturing equipment to make Lorabid because they were expecting Lorabid to be like C-Clor, a big seller, but it wasn't. It was kind of a flop. So basically, they tore that facility down, and then they decided to basically move the development people off of that site. So then we moved down to Indianapolis, and then we were there about 10 years before the layoffs.
What drugs did you start at when you moved downtown?
Probably the biggest one that I worked on was something called arzoxafine.
Arzoxafine?
Arzoxafine, it was for
osteoporosis.
The
serum.
Yeah.
Okay.
So
okay.
It was basically a next generation Evista.
Raloxifene.
It was designed to be much more potent in all of the assays. So it was a much more potent molecule. And we put that into the clinic, and Lilly spent tons and tons of money on that. It was in the clinic like 4 or 5 years. And at that time, FDA was asking for bigger and bigger trials and more data. So they spent, many, many millions, hundreds of millions on that. At the end of the phase 3 clinical trial, they unblinded the data, and they basically found out it wasn't any better than the standard of care. So they decided not to commercialize it.
So did you have a trial of like placebo control, or was it was
I think it was
compared to Avesta?
Compared to Avesta and some other standard of care. So it wasn't any better than the Avesta.
You said it was more potent.
Yeah,
It
was more potent in all the laboratory assays.
But it didn't produce any better like bone density.
Yeah, it was for spine fractures, to prevent spine fractures. So it wasn't any more effective at preventing spine fractures than Avesta.
What about the safety data? Since it was more potent, did it cause more like clotting? Or anything?
It was safe. Safety profile was very good. It was a good drug. So yeah, I spent a lot of time on that. I actually wrote a lot of the submission for that, the CMNC portion of the submission.
So it was basically a Me Too drug that didn't.
Well, not a Me Too. It was a next generation. It was designed to be much better, but it didn't turn out that way in the clinic. So that's part of life and drug development.
What was the next one you worked on?
Then I moved from Lafayette down to the Indianapolis site and worked on a molecule called CDK4/6 inhibitor.
Oh, it's a breast cancer drug.
Yeah.
Okay.
It's marketed as Verzenio.
Verzenio. What's the generic?
Abemaciclib is the generic name.
Okay.
Yeah, I also worked on some other drugs up there, the olanzapine PAM08, Zyprexa Depot, and also Strattera. Did a little bit of work for Strattera.
Yeah, Strattera must have been a big one. Because was that like the first NRI for ADHD?
Yeah, I'm not sure. I just did some. That wasn't my main project. I just was kind of helping on that project. So we had a question from the FDA on our submission. They had a question about a certain impurity. So being an analytical chemist, what I would do is come up with a method to check for that impurity to make sure it wasn't present.